Good afternoon, everyone. I'm thrilled to bring to you the inaugural seminar for the USC Norris Center for Cancer Drug Development, a collaboration with USC's Mesh Academy. We're really thrilled today to have Dr. Michael Jackson, who's the Senior Vice President of Drug Discovery and Development at Sanford Burnham Prebys Medical Discovery Institute, heretofore known as SBP. Dr. Jackson oversees the Prebys Center, which is approximately 60-person academic drug discovery enterprise that's embedded within the Sanford Burnham Institute. The center has state-of-the-art, ultra high throughput drug screening and lead discovery and optimization capabilities. And Dr. Jackson's overall goal is to generate a pipeline of first-in-class small molecule therapeutics based on the breakthrough discoveries on the molecular basis of disease made by investigators at the Institute and other collaborators. Upon joining SBP in 2009, Dr. Jackson managed and then led as principal investigator SBP's NIH-funded Molecular Libraries Probe Comprehensive Center, which successfully executed over 100 high-throughput screening campaigns with collaborators across the nation. In fact, over a 6-year period, the center generated greater than 30 million screening data points and produced over 60 chemical probe reports published through the NCBI bookshelf. Since the Molecular Libraries program ended, the Prebys Center has continued to operate in a highly collaborative framework with investigators from many academic institutes and biopharmaceutical companies, and it continues to conduct high-throughput screens against 20 different targets each year in search of drug leads to advance the therapeutics. Dr. Jackson's integrally involved in the center's outreach and translational efforts that include over 35 different ongoing collaborations, most of these supported by NIH or DoD grants or through contracts and collaborations with biopharmaceutical companies. And we're hoping to establish stronger collaborations with Prebys as well. I'll turn it over to Dr. Jackson, who will talk about accelerating oncology therapeutics into the clinic through collaborative drug development. Dr. Jackson, thank you so much for joining us today. And there we go.
That work?
All good, yes.
All right. Well, thank you very much to provide me the opportunity to tell you a little bit about what goes on at both Sanford Burnham Prebys, and in particular the Prebys Center, which was just mentioned is really an academic drug discovery center looking to partner with people to move forward ideas and concepts that you may have of a new drug. I'm really focused primarily on small molecule drugs rather than biologics. That's really our platform. And let me tell you a little bit about it. Before I do, let me just say a little bit. Why can't this work? Okay. Just at Sanford Burnham Prebys, it's really got 3 elements to it. Basic research, fundamental research. It's not a hospital. It's a bench laboratory-based organization looking to do medical discovery. We've got the therapeutics discovery, which I'm going to tell you about, the Prebys Center. And then, of course, we have a graduate school, and we do a lot of postdocs. So overall, the Institute, for those, it's just down the road from you guys in San Diego, right opposite Scripps, and right in the hub of on the Torrey Pines Mesa. And it's about 600 employees, about 45 faculty labs, nearly all of which are really funded through NIH fundamental grants underpinning the research. Top of the list of the centers that are here, we are one of only 7 NCI designated basic research cancer centers with a cancer center support grant. And that really cements the Institute's focus to cancer, and hence I think a lot of interest and overlap with research ongoing at USC. In addition to these centers, as you can see, we have the Drug Discovery Center, as just mentioned. This is called the Prebys Center due to a donation from a philanthropist, Conrad Prebys, who really was looking to accelerate and help us advance academic drug discovery. And so the center has about 55 employees today. About 60% of those have a PhD. Many of the employees have been trained or have spent time in industry. And we have, as you can see here, this sort of flower, the Prebys Center flower, all the components, elements that really allow you to start from a concept of a drug all the way through doing assay development, screening, various different other ways of trying to find a chemical hit or starting point for a drug. And then we're going to move on with medicinal chemistry and pharmacology and whatever tools we can have to try and advance that through to at least what we would call the lead. And I'll tell you a little bit more about all of this in a moment. But this is why it's sort of a one-stop shop to come to try and collaborate with if you've got ideas that you want to advance from a translational perspective. So here's some mugshots. It's a bit of an odd-looking crew, but there you go. That's who we are. This just shows you the main leadership team. As you can see, composition of biologists, chemists, people with expertise in assay development, pharmacology, and even stem cell biology with Ambang and her experience of making stem cell derived models. This crew has, as I say, a lot of industry experience, and collectively really are the drive behind our attempts to work with faculty from here and from elsewhere to drive forward new ideas for drugs. So this is kind of the overall game plan of working together to build a pipeline. And what I'm going to try and do today is to walk you through some of this. And in particular, you can have lots of great ideas, but if you don't have any funding to move them forward, they're just good ideas. So really, at the end of the day, we kind of have tried to identify how we can move forward our ideas from basically the very front end here, which is to identify targets. And I'm sure many of you have ideas of a target, a potential drug target or a pathway or a process. And then we're going to try and the expectation is that you're going to validate that by some means that indicates that knocking it out, down, increasing the levels of it, or whatever modulation you can imagine, actually is going to have a therapeutic benefit. And with that, we would then start to come together and start to develop assays to conduct high throughput screens. And then from those screens, which will find hits, those are hits from a chemical screen, the large chemical library screen. And then we're going to move those hits to leads, leads being the lead to a drug, mainly using medicinal chemistry and some driving assays, and then moving those leads forward through a lead optimization process, which is heavily reliant on medicinal chemistry and pharmacology, and move those towards compounds which are suitable to test your concept in a gold standard animal model or a cellular model, an organoid model, or whichever model you wish, that then tells you that this whole thing looks like not only is it a druggable pathway, it's something that actually is going to do more good than harm and has a therapeutic utility. So the Prebys Center in particular is really focused on the development of the small molecule. So if we look to advance this across these various different steps of stages of assay development, screening, hit to lead, etc., this is an expensive process. And even the very beginning stages of this, just to say I have a target and I want to develop a high throughput screen, and then see if I can find some compounds that legitimately bind selectively to my target, that's $1 million experiment to do properly. So our institute doesn't have millions of dollars lying around to do that. What instead we've been doing is what we would call grant leverage strategy. The NIH and other DoD and other organizations have funds to help conduct these translational activities. And they're grants of assay development grants, high throughput screening grants, even pro-discovery, lead discovery grants, and in fact, even grants that would take your drug lead all the way to a phase 1 clinical trial. So we have focused our efforts over the last 5 or 6 years really to try and get these grants so that we can move our pipeline forward. And so what I wanted to do today a little bit was to inform you a little bit about these grants if you don't know about them. Secondly, to tell you kind of how to move, what's expected and how does this all work. We at the moment have about 40 active translational grants, and you can see supported by over 20 R01 grants and various DoD grants. And some of these, even these UH3 and U01 grants that allow you to actually advance projects all the way from an optimized lead actually through preclinical IND enabling studies to the clinic. So we have a lot of experience in doing this. This is about $15 to $20 million of grant funding a year that is supporting our portfolio. So I'm sure that some of you are familiar with these, but I guess is that not most people. Most people don't think of an R01 grant really being to support that type of activity. So I thought what might be useful would be to talk a little bit about some of this, the types of grants that are there, what sort of research or translational research they support. What are the various opportunities, the sizes of those, the duration and the success rates. Just in general, what's the data you need, what scores well, what are the components. As I say, we have a lot of experience in this zone, and you don't have to partner with us, but I think you would, we are a reasonably good partner for people who are looking to advance translational objectives through grant funding of some type, form or another. So I don't, there are 20 to 30 translational grant opportunities out there from the NIH. Here just shows you a few of them. I'm happy to help anybody if you've got specific interests and figuring out whether your particular idea is suitable for a grant. So this is the NIH ones. On the one side here, we've got grants that support mainly assay development. You see these are mainly R01s and R21s. And on the other side here, hit to lead optimization grants. These are again R01s, R21s, R61 leading to an R33. And I haven't shown up here, but there's also the blueprint initiatives and the U01s and the UH3s that can also apply for a later stage. So at the very beginning of this, in a way, there's a sort of a division of labor that goes on, or a division. First of all, the Prebys Center, which I run, this drug discovery academic center within Sanford Burnham. We're not a for-profit organization. We are absolutely an academic organization that conducts more activities are more typically thought of as occurring in biotechs or CROs. Our role really at the end of the day is, once the idea of what sort of a drug we want to develop, our job is to develop the best possible compound that we can, right, against that. The other side is the people that we typically are partnering with, they hold the keys to the idea. So the target validation, the therapeutic hypothesis, who are the patients that are going to benefit? What are the animal models, the cellular models that really tell us we're on track? So that's kind of the first thing. And this is really critical because the therapeutic hypothesis, and it really talks to how important and how significant this work is likely to be. And I'll come to that in a moment. And so while this is a very busy slide, it shows these two kind of activities across the drug discovery process, as I say, where we start with a project and then try and find a chemical hit, i.e. through high throughput screening, and then convert that into a lead to a drug, optimize that lead such that it's suitable for testing properly in animals. And that then, if that starts looking encouraging, look to the next final stages of actually polishing that to become a drug, and then the IND enabling studies to go to phase 1. In the top of this line here, where it says concept, assay development, high throughput screen, hit to lead, early lead optimization, that is really the activity that the Prebys Center is going to focus its efforts on. The collaborators are really focusing on target identification and validation, and then building the really bespoke models the cellular models that are really reflective of the biology we're trying to target, and then the in vivo models, which are, of course, critical, along with really trying to develop the biomarkers that are going to be used in the clinic to tell us that we've got on-target activity and that we have engaged the target and we're making progress. So not going through all the rest of this, these are just the steps and stages, but just to say that that's kind of the division. And as we move on to how are we going to get this funded, these translational grants, like all NIH grants, are highly competitive. And interestingly, although they are of a slightly different nature than a typical R01, R21 grant, they still have the same components. In other words, they still get scored on significance, investigators, innovation, approach, and environment. So let's go into a little bit of that, because I think as to whether one of the ways that we try and identify what we think the best projects to advance are is, do we think they're going to score well so that we can get a $1 to $2 million R01 grant to advance the project from concept through to a validated hit or an early lead. So of all of these things, it has to be said that the one thing that is difficult to overcome is if the study section doesn't like your therapeutic hypothesis and thinks it's very thin. That's hard to recover from, right? Doesn't matter how good you are. So no pressure, guys, but at some level, the therapeutic hypothesis and the strength of that and the strength of your target validation and the underpinnings of the project are critical. Of the next things that are important and are things that we know we can score well on and have routinely scored well on are things like environment. So so that's the NIH scoring. There is another program that is more of a contract-based process, which is run by the NCI. This is the next NCI Experimental Therapeutics program, of which there is a chemical biology consortium. And this, you can see how they are rating things. Again, scientific merit. The top of the list is the hypothesis supported by the current state of the field. And so really trying to narrow it down to, do we really believe the therapeutic hypothesis? So coming back then to the translational grants from the NIH, one area that I want to tell you a little bit about, because it reads into our translational capabilities at the Prebys Center. And so I'm going to walk you a little bit through components of innovation and approach. But first, let's talk about environment. And then particularly going to score how strong our research capabilities are, how strong our drug discovery capabilities are, and in particular on the investigator, how strong is the experience of the drug discovery team. And that's a place that we score well. And it's not surprising we score well, because the Prebys Center has a good deal of experience in conducting this type of activity. And as you can see here over the last 10 years, you can see how we have really been conducting, well over, some years, 20, 30 high throughput screens a year. And we're making a few thousand compounds every year. So that shows you our strength in medicinal chemistry and to move projects beyond having found things as a hit and to take them to a lead so that we can test our therapeutic hypothesis in animals. We have a great toolbox of capabilities, all sorts of different assays, cell-based assays, biochemical assays. I don't have time to go into all of these. They're on the website about the Prebys Center, and happy to discuss them further. Some of these are pretty cutting edge, qPCR, and I'll tell you a little bit about that later. And some of our high content imaging is pretty cutting edge. Other things are just, enzyme assays are enzyme assays, but you've got to set them up properly. So these capabilities, along with the fact that we have a chemical collection of nearly 1 million diverse compounds, puts us in a strong position. And those compounds are in various different flavors. We've got diversity, and we also have focus libraries. And the focus libraries contain specialized libraries around particular protein families or activities like epigenetics, kinases, but also we have nearly a complete collection of the FDA-approved drugs, along with many phase 1, phase 2, phase 3 drugs, so that we can look to explore, particularly in phenotypic screens, what their utility might be. So all of these libraries, of course, require that we can screen shown over here on the right-hand side. You can see a little movie of our HRB system. It's a fully integrated robot that can easily screen 100,000 compounds a day and can do more, but that's about the pace we normally go at. We have screened many targets using this, and shown in the pie diagram all the various different reporters and readers, and including state-of-the-art high-content imaging using a Phoenix Opera, and I'll tell you a bit more about that in a moment. So this robotics allows you to basically be responsive to a grant looking to explore a first-in-class target and trying to find chemical probes against it. Also to say that the automation is equipped with acoustic dispensing. So this is where you're using sound waves to move liquids around. And you move liquids, you can show the little pinging little droplet of, that's a 2.5 nanoliter droplet using an acoustic wave that actually pings liquid from one plate to another, giving you truly touchless, tipless dispensing with absolute precision. And once one starts using these machines, and we have 4 of them, you become pretty much obsessed when you really don't want to go back, because your error bars go down and the precision goes up and everything is good. So that's kind of how we move liquids around primarily in our automation. And so that's kind of on the environment side. We do well for obvious reasons. We've got lots of technology, platform, and people. The next thing that really is going to make a difference is in getting a high score is innovation. Now, it can be innovation just simply in the target itself, but all too frequently would like to see innovation in how you're actually setting up your assay, in terms of how are you going about doing this? Is it innovative or is it ho-hum? And so I want to tell you a few components of innovation, and particularly around what I call, from the front end, where I think many people's projects start at, is assays that find real hits. So I'm sure everyone is familiar with, generally high throughput screening at some level. These are conducted in 384 well or 1536 well plates, so miniaturized examples, and where people are trying to put in reporters and various other things that actually can enable high throughput screening at a reasonable cost. We have had a lot of angst over many of the assays, and I'll come to that in a moment. But I want to highlight a few assays that I think you might be thinking, how would I tackle my protein? I wanted to just go through highlight a few of these. I won't go through it in too much detail, so I apologize. It will be rather a rapid blast through it. But nevertheless, hopefully you'll get something out of it. So this is particularly the section called balancing throughput and relevance, right? So at any one point, the more you dumb down your assay and your screen, the further away it gets from being physiologically relevant. And so therefore, the more and more chance you have of discovering chemical matter that is irrelevant, actually ultimately to modulating real biology in real cells in a real organism. But having said that, there are certain technologies that we have become very enamored by. And I want to go through a couple of those, particularly protein thermal shift. And that's actually more recently being followed up with a version of that that's actually cell-based. Then talk about 1536 well qPCR in 3 colors rather than having reporters, and then move into my final example on phenotypic screens using primary human cancer cells. So here's one program. This is PHGDH. It's a cancer metabolism target. It's the critical enzyme in serine metabolism, and is well known to be upregulated, amplified, and various other things in many cancers, particularly neuroendocrine prostate cancer. So this is a project that Jorge Moscat, who was a professor here at SBP until recently when he moved to Weill Cornell, really was leading. It was a project that was selected by the NCI Experimental Therapeutics program and was funded for 4 years to tackle this and see whether we could find a first-in-class drug against it. And I also showed the other 2 collaborators, Edward Sajenka, who really pioneered many of the assays, and Brad, who was the medicinal chemist behind this. Now, here we had, I'm not going through all of this, but we had an abundance and almost an embarrassment of riches in terms of the number of possible assays that we could conduct. We had obviously enzymatic activity because it's an enzyme. We also could do binding assays. But the one I want to highlight, and the one we chose for this program, was to lead with protein thermal shift. Now, protein thermal shift is a technology where you purify the protein, if you can, assuming that you can purify it, the protein of interest, in this case, an enzyme, so you should be able to. And then what you do is you purify it in large quantities, and then you put a small amount of the protein in the well of a 384 well dish, and then you heat up that protein, and as it heats up, it denatures, and portions of the protein that otherwise are hydrophobic and buried within the folded protein pop out and bind a dye, Cipro orange, and so you can monitor the unfolding of the protein. And this can be done in a PCR machine, and it remarkably consistent profile of how a protein melts and how it, and so the goal here is, or what's being realized is that if a small molecule binds to your protein, you will change the temperature at which the average molecule unfolds. And so it's a very simplistic way of looking for compounds that bind your protein. Now, that doesn't mean they do anything. It just means they bind. So we have applied this to many, many, many proteins, about 50 proteins at the moment. Really like this technology as a leading technology to find first the chemicals in our library that bind the target, and then downstream of that, we're going to start looking for other assays that we can have. So you're going to do the initial 300,000 compound screen using protein thermal shift. So looking at how each of those 300,000 compounds can thermally stabilize, or in some cases destabilize, a particular protein. It's amazingly accurate. It can detect different, within 0.1 of a degree, the compound binding. We typically look for compounds that shift the melt curve by about 1 degree. And then downstream of that, you're going to have counter screens with a counter protein. And then we're going to start moving into other assays, like does it affect enzymatic activity? And does it affect, can we show that it binds something in a cell? So here shows you kind of how this sort of works. We're going to do a screen, and we're going to positive controls, negative controls. And you can see here, the positive control gave a very nice shift here. And this is a very nice sort of distribution of screening hits. I'm not going to go through it, only to say on the far side here, what you start to get is a thermogram. You start to see that certain compounds, as you increase the concentration of them, will give you a nice dose proportional temperature shift. As you can see in the first case, 689 and 096, they're giving 1 to 2 degrees. And we have gone on to show that these were our initial screening hits. Not only did they hit the enzyme in terms of altering its thermal shift, you can see on the right-hand side in the next panel, you can see they also started to inhibit the enzymatic activity. So that's pretty good. So we've got a compound, thermally stabilizes the protein, and starts to show us evidence of inhibition of the enzyme. You can see there are other compounds down here that actually caused a thermal shift, but really did nothing to the inhibition of the enzyme, suggesting that they bind somewhere else. And in fact, that's the beauty of this technology. You can find inhibitors, activators, allosteric modulators, all sorts of things. It's a frontline assay. So that's kind of the beginnings of how we started this program, finding hits and moving them forward. The next thing we wanted to ask is, well, how do we know that it looks like this is a good compound? How do we know that it causes thermal stability to the protein, it inhibits the enzyme, but does it work in a cell? And this is complicated, and I would encourage anybody who's interested in assay development to really look up this technology. It's a version of thermal shift, but it's actually within a cell. And so the way this works is you take your protein of interest, you tag it with a little 11 amino acid tag called a high bit tag from Promega. This tag is the complementation fragment that matches another part of the thing called large bit. So that's a luciferase, and which is inactive unless the tag is present. So the way this works is you tag your protein of interest over here. You then transfect cells with a plasmid encoding it, or you can even put the tag in by CRISPR if you're more excited, if you've got more time. You then put that protein, you take the cells, you put the cells in the wells of a 96 or a 384 well, and then you have a temperature gradient for 3 minutes. And then what you're going to do is you're then going to lyse the cells and see, okay, how much of my high bit tag is available? So ordinarily, as you heat the cells up, the high bit tag is going to be occluded by the protein denaturing. So at some point, the protein will denature and the tag will be occluded and will not be able to be complemented. So when you add the large bit, the large bit will be inactive and not be able to take the substrate from furazine to generate light. All right. Whereas if you add a comp, so that's your baseline. And then amazingly, I have to say, I thought this technology would be way more ragged than it is. When you add a compound that binds, and in case like the one I just told you, that 096, it caused a significant shift in the temperature at which the protein actually denatures. So ordinarily, the protein would be fully denatured. With the compound present, you get a 2 to 3 degree shift in how far and in the protein still staying in solution, and you can detect that. And so what that looks like then is, here are some compounds where we've done the curves, and you can see quite nicely here that certain compounds are nicely stabilized, other things destabilize, so you get various flavors, and you get that actually showing you that not only does the compound get into the cell, but it influences the target of your interest. So I think that's really very exciting technology, and we are now applying this to almost every project we can. This particular one of these, which was a strong destabilizer, actually went on, and we actually were able to co-crystallize that compound and go on to do medicinal chemistry, and you can see with time get to a pretty nice compound that now is 76 nanomolar compound being driven by structure-based drug design. This program is hit some snags, and we're struggling with it again for other reasons, but I think it's a good example of how you can of a testing funnel leading to some potent leads. The next, so the key components of thermal shift are that you know, as long as you can make your protein, it's well folded, and you can make quite a lot of it cheaply, it's a great technology. We try to avoid any tags on the protein, or remove them at the end. And interestingly, it will also work for protein complex, which is kind of fun. Works on, we've been using on kinases, phosphatases, proteases, enzymes, you name it. And most excitingly, you can find activators. So with a project that we worked on with colleagues at Daiichi Sankyo, we were able to find activators of the NAMPT enzyme, which I thought was a pretty high bar, but we got some very potent, nice activators using thermal shift as the leading technology. All right, so let's move on then to another innovation that would be assays that monitor transcription in primary or primary human cells, cancer cells. So we in the center have run many luciferase reporter assays, and frankly, I don't want to run any more. And part of the reason why is that they just are plagued with false positives that seem very difficult to screen out. So the noise can overwhelm the signal in a screen. It does not mean that the assays don't work. They do work. They're just not particularly good in our hands anyway for finding initial hits from a large chemical screen. I think they're also compromised fundamentally because the native state, obviously, of the promoter is affected in general, unless you CRISPR things in. And from our experience, we have done many and never really found a project in which we've used this that has really gone on to a late lead or to a drug. So we were excited by the use potentially of qPCR as a screening platform. This is being troublesome. It's expensive and difficult to do, but I want to show you an example of one where we've done this most recently, and two, how we're now moving this. Initially, it was just looking at one RNA transcript, now to many, well, to 3 or 4, so 4-color qPCR. And of course, the advantage is here that you're going to directly monitor the RNA levels. There's no transfection, no selection. You can go straight from primary human cells straight into the experiment with no intermediate reporter. And of course, this takes up the physiological relevance of the screen. So this was an experiment that Sumit Chanda, who was interested in finding activators of the CGAS STING pathway. And as you know, there's interest in that from a vaccine adjuvant side, but also from immuno-oncology aspect of whether you could find something that would be useful to make cold tumors hot. So the way this worked was did a lymphoresis, then took monocytes from the patient's or the donor's blood, differentiate those into monocyte-derived dendritic cells, put those in a 384 well plate, add compounds into the various different wells, Then we're going to make RNA from that. Then we do qPCR, and we're going to actually do looking at transcript. In this case, we're looking at ISG54. It's a gene that was well known to be significantly upregulated when these pathways were switched on. So this is somewhat of a phenotypic screen. Technically it was tough to do, but we got through it. This was the first time we'd done it. We got a reasonable hit rate, and we started to find compounds immediately that were reasonably potent, to be quite honest. A micromolar, 1.8 micromolar, 1 micromolar on cells. We have moved those forward now and moving those towards animal models. And they're showing some activity there. So I want to bring this up because I think many people are interested in finding compounds that switch off or switch on transcriptional programs. I'm particularly excited that you can do that directly using primary cells or primary cancer cells or whatever you want to, the real deal. And we are getting so we can measure up to 4 RNAs at a time, allowing you potentially to be able to look at a transcriptional components of a transcriptional signature, 2 genes going up or 2 RNAs going up and 2 going down. So it is to be said that in general, it's better if you're working with reasonably abundant RNAs, and the downside is still that it's pretty expensive. Nevertheless, I think a very exciting development that really could allow us to find many more relevant hits to really important targets. The last bit of innovation I want to touch on is phenotypic screens. This is probably much more obvious to people. The use of high content imaging to be able to monitor and measure various different elements in 2D, 3D, all sorts of things that one can measure. The Prebys Center, this is one of its real strengths. When we were a molecular libraries program comprehensive center, this was our differentiating technology. And we've gone on to kind of buy all the bits and pieces to make this happen at reasonably high throughput, including all of the informatics behind the scene. Not only do we have the assay plate preparation and all those systems and the liquid handlers, but then of course, taking the information from those and then digesting it and then being able to feed it all to the user so that you can find the compounds. I want to use an example here, something from Robert Westwood-Rea, who's a PI at the Institute here. He was particularly interested in a rare childhood cancer, medulloblastoma, and where transcriptomics had been done in 2010 that showed that there are 4 major groups of medulloblastoma: Wnt, sonic hedgehog, group 3, group 4. Group 3 was the one he was particularly interested in. It's the most deadly. And that signature pattern was known to really be generated due to genetic amplification of MYC. And so MYC was the target. And so how are you going to tackle this? And so what he encouraged us, and we worked with him, we were going to basically go take material from the resected tumor from the brain of a child, passage it through the brain of a mouse, take those cells straight from the mouse brain and put them straight into a 1536 well plate. And then we were going to put compounds on and then use an antibody to detect the MYC protein from these PDX cells. I have to say, I thought this was completely technically on the edge. Happy to say that Suzanne Hainan, who's the director of our high content microscopy and screening operations, was able to wrestle this to the ground. And we were able to conduct this screen. It was put the compounds on for 4 hours and then look for compounds that will reduce the level of MYC protein in that period of time. This is a bit of an overcomplicated slide, but again, to say that despite we screened 100,000 compounds, looking for compounds that would decrease the levels of MYC, leave the levels of RB unchanged. And actually we were not looking at, we didn't expect to have things that would be overtly toxic at 4 hours. We were able to find 15 compounds, 3 scaffolds, and one of these compounds has gone on to be pretty spectacular. Note though that this whole thing took us 7 months to conduct, and yet we still got a very nice reproducible assay. And if you're into Z-scores, it's 0.59. Very impressive reproducibility for something in which you've gone through a mouse brain. So here's an example of a hit profile. You can see that it reduced the level of MYC protein at 11.7 micromolar. Didn't really do much to RB or cytotox. This is what the reader was reading. And in fact, this project now has gone on to get an R6133 Ignite award from NINDS to advance it. And here shows one of the more recent compounds, 7047, And you can see we're now down to an IC50 of around 200 nanomolar. And in fact, the most recent compounds are now down to 50 to 20 nanomolar on cells. And we're hunting down the molecular mechanism of these, but I think it shows you the power of that technology on primary human cells. So again, phenotypic screening, the take homes here, use the most relevant cells you possibly can, ideally human primary cells. We like to track the natively expressed protein with an antibody if you possibly can. If not, you can put some tags in, but we would prefer to CRISPR in the tag rather than try to have overexpressed proteins as surrogates. In our experience, these assays work best when we have what I call smaller, grayer boxes rather than black box screens. So where you are making something happen in a short period of time. So in this case, degradation of a protein in 4 hours due to the fact we added something. So we triggered something to happen, and then we monitor that process. So good to find degraders. And our experience has been that these assays have typically very low hit rates, probably reflecting the fact that the assay is really accurately reporting effects on the cell on the real biology. So that was the innovation. The last component I also want to touch on is the approach. And approach means all sorts of components in these grants, but in particular, it really seems to be, and I'm going to talk particularly about PAR 2271, which is the screening for compounds for drugs, immunomodulators, or for high throughput screenings, things like these. And this is where you have to describe your complete critical path testing funnel. So you're obviously going to have your primary screen, your hit confirmation, all the components downstream. You're trying to, again, balance this throughput with relevance as to, how big a library should I screen? Should I focus it? What's the best primary screen? What's the best secondary screen? All these things and components you need to put together. And most importantly, you have to have, for a grant, you have to operationalize this. And what does that mean? It means that you need to have conducted a pilot screen showing that the performance of your primary screen in a high throughput format, 384 or 50, is suitable. Secondly, you have to show that you have in place the counter screens so that you can prosecute the hits that came from your pilot screen. And you really should have the other downstream assays that allow you to go from primary screen down through validated hits. So something that you believe is absolutely selectively binding your target, or all in place. So that's the preliminary data that you need to put together. And most of these grants have typically 3 aims. They have the critical path diagram, a chart that shows who's going to do what. So these charts are typical Gantt charts. They'll sort of have the various different components of scale up of the screens, performing the validation, and then characterization. So those are the sorts of components that you can expect. So hopefully in this, hopefully I could tell you at least some of the components of these translational grants, what sort of research they support, the opportunities, the data, the components, and what would sort of be expected. So with that, I think I shall end up just thank all my colleagues in the center. Is the motley crew of us from days when we were allowed to not be so socially distanced. And with that, I'd be happy to take any questions.
All right. Thank you, Michael, for that really elegant presentation. I learned a lot. And we have a question here that is, what are the mechanisms of actions of the compounds you're identifying that inhibit the activity of transcription factors?
You don't necessarily know, right? So, that screen I just told you about, for example, was, if one is referring to the PCR-based screen, it's a phenotypic screen, and then you have to work backwards from that screen to try and figure out what the mechanism is. There are different ways to try and deconvolute, if you want, from phenotypic screens, and that's a general discussion. First of all, I think if you keep your time frame relatively short as to how long you conduct your screen for, so in the transcriptional things we were typically doing, it depends, 3 to 6 hours is a good period of time, so that there aren't so many things that could have happened, so that your compound and however it worked was like 5 steps back. I'm not answering your question because like any phenotypic screen, the exact mechanism of action is requires a lot of workup, and even then, it's a reason why many people will screen bioactive compound collections in which the compounds are pretty well annotated in the first place as to what they hit, to allow you to shortcut the process of target identification post phenotypic screen.
Okay, thank you. And we're seeing lots of additional questions come up in the Q&A section. So another question, investigator would like to know more about go and no-go decision making at the different steps of drug development. How do you decide what are the most important factors, and do you have a review committee?
Well, it's complicated, I would say. The reason you want to, so one always starts out with the best of intentions, and which means that for every step and stage, you pre-agree to a set of a profile for the molecule that you are trying to generate. And you say, and when we do this, it's a pretty comprehensive and exhaustive list. And obviously the further you move along the drug discovery and development cycle, the longer that list becomes in terms of the properties, because it starts getting to safety and bioavailability and all sorts of other things. So we normally have a series of elements that are must-haves, non-negotiable. And then we have another series of nice-to-haves, where I would prefer it didn't hit the hERG channel at all, but if it hits something a little bit, maybe. And so we then have a committee in the sense that a number of us, and you saw some of the experience set in the center, we use our experience to juggle all those things together as to whether an issue that we have identified where our compound isn't quite as good as we would like, is it a showstopper, or is it something we think we can solve a bit further on? So I'm not really answering your question again, but I would say that's where you want experience, because it's not a box sticking exercise. And in fact, it's very dangerous to get over prescribing of exactly what your drug will do. Drugs are very rare chemicals, right? That they do more good than harm sometimes in the right person, right? And so you need to have a deep understanding of the patient too, I think, as to what would be tolerated. Yeah, I think experience is what you really need. You need some people who have done it before and know what is really a showstopper developmentally. So you really can't develop that. You're going to have problems with the regulators, and you just wish you never started. Basically, it's just going to be such swimming against the current, right, for such a lot. Whereas other things, that's theoretical, and I know on paper it doesn't look so good, but that's just still theoretical until we actually have that data that shows that's true.
Okay, and just a follow-up to that from me. So would you consider commercialization potential a must-have, or would be nice?
Well, that's an interesting point. So the NIH, although they are so let me tackle that 2 different ways. And this is actually a very interesting point to it. In a way, the NIH really is focused more on innovation than so much commercial viability. And so it has become one of those areas where people have angst around it, where you can get a grant, but they say, well, I'm not sure you're really discovering a drug, you're discovering a chemical probe. And I think some of that criticism is fair. I think on the other hand, is maybe a misguided. Maybe we know quite a lot about biology, but we don't know everything. And in fact, chemical probes and tools may lead us to something that was unexpected. So we at the Institute right now do not have commercial viability of a project being the first hurdle to get through. We have more, we may change, but we've more been, do I think this could attract a translational grant, right? And if the answer to that is no, I don't, then you better come up with some other method of funding it, because otherwise it's just an idea.
Yeah, I happen to agree with you, so thanks. Okay, so, this is from your friend Steve K. Great talk, Michael. He said the PTS screens look really interesting. Is there a high dropout rate of hits due to bioavailability or physiochemical properties, agonist versus antagonist, etc.?
I wouldn't say. I mean, it is a biophysical, if you want, screen. I mean, it's what I didn't explain so much about it. One of the reasons we particularly like it is it's what we call a mass action screen, and so you've got about a microgram of protein in each well, and you are putting compound into that well. And as we all know, our compound collections aren't perfect, but to move to shift the thermal stability of 1 microgram of protein, this cannot be a minor contaminant of chemical in that well, right? And so the only way you can shift the whole microgram of protein is that it is the abundant chemical in that well. So we like it for that reason. Whether those compounds fall out later because they're not cell penetrant or this or that, that is obviously a concern. And that's why I think the CETSA assay, well, we're not allowed to call it CETSA, but the thermal shift that's in cells, I think CETSA is patented, so I have to be careful what I say, trademarked or something. But I think that's another extremely interesting technology, right? And I was super surprised that it's so reproducible. You could see some of those curves there. They, you know, who was to think that technology would be able to, within a, half a degree, reproducibly give the same sort of denaturation temperature every single time in a cell, and such that it was sensitive to the binding of a compound. I'm surprised, actually.
We have a couple more. So someone seeking your opinion. It's easy to validate a compound. Is it easy to validate a compound without knowing the mechanism if it's not toxic and has good solubility?
Is it easy? Say that first bit again. Is it easy to?
Yeah, I'm just reading it. But what's your opinion? Is it easy to validate a compound without knowing the mechanism?
So let me give you I mean, if I go back to that example of the MYC degrader, right? We don't know the mechanism. But what I can tell you is that MYC protein levels are the clinical biomarker. So are we trying to find the molecular mechanism? Sure we are. Are we trying really hard? Yes, we are. But do I believe that compound, which is now operating at about 20 nanomolar on cells, causing nearly a 95% reduction in MYC levels in 2 hours, do I believe that could be a drug? Yeah, without me knowing how it works. Now, will I be more comfortable if I do know how it works so I can better understand mechanistic talks? Sure I will. But there are plenty of drugs on the market that we don't know how they work. And in fact, some of the ones on the market, we think we know how they work, they don't work that way. So before we all jump to conclusions that the world is squeaky clean, I think biology is messy, and particularly small molecule pharmaceutical, cleanliness is perhaps overrated. I'm not sure if that helps, but that's kind of my stance on it a bit, I think.
Yeah, that is helpful. Do you have any insights into developing high throughput screens against membrane-bound targets?
Sure. Yeah, very much so. So my favorite targets are membrane-bound, obviously. G-protein coupled receptors. Interestingly, when we, doesn't know that it has to be a G-protein coupled receptor, but it could be a receptor tyrosine kinase. Plenty of great drug targets that are on the cell membrane. I would only say that if something is on the cell membrane and it can be attacked by an antibody, not attacked, but it can be addressed with an antibody, and that antibody can do the function you want, that generating antibodies is more of an engineering world than the discovery of small molecules. So going back to, we do lots of cell-based assays looking, whether they're against G-protein coupled receptors, ion channels, cell surface membrane proteins. All of those are well in scope, whether they're using high contact microscopy or whether they're using other techniques. calcium imaging, all sorts of other cell-based assays I didn't get into. And I didn't go through the whole toolbox of assays because I just wanted to highlight a few that I thought maybe were a little less familiar to people in the innovation section.
Okay. I'll add another question. Do you, particularly as you move toward the later stages, do you take a deliberate approach of considering companion biomarkers? I mean, you mentioned, you know, protein biomarkers. Do you try to develop these in tandem, or?
We do, but as I hopefully pointed out, that in general, although, so the most advanced project we currently have is a tissue non-specific alkaline phosphatase inhibitor. It's in the clinic with Daiichi Sankyo. And we were able to devise an assay where we were able to just simply take blood and then determine the extent to which our compound dosed orally was actually inhibiting the alkaline phosphatase in the blood directly. So you have a completely PK/PD relationship that you're able to do in a patient in a phase 1. In fact, that's what they did in a phase 1.
Right.
Now going to cancer, of course, it's even more important to be able to define all of what you would like to have up front. In general, I say that, as I try to indicate this sort of division of labor, it's not that we will absolutely help technically develop that assay, but obviously the therapeutic hypothesis and the underlying biology and thinking behind what is the right biomarker is more likely to come from the clinician / the expert in the biology than it is from people who are more generalists in developing drugs.
Okay, thank you. And let's see. Okay, well, the last comment was a thank you, fantastic talk. So maybe that's a good place to close as we are about at the hour. Michael, thank you so much.
Oh, you're welcome. Yeah.
Really terrific talk, and we look forward to building some collaborations with your center and with you.
All right, very good. Well, thanks for listening. Yeah.
Thank you. Bye-bye.
All right.